Hi there. In this video, I'll be describing the replication fork that forms during DNA replication in prokaryotes. In a sense, I'll also somewhat be describing the process itself in order to show the functions of different enzymes during DNA replication. To start, we need to unwind DNA, as this is how we can create the space needed to form a complementary strand of DNA or replicated DNA. To do this, we have what is called the origin of replication, where an initiator protein will bind, causing a double helix to unwind with the help of DNA helicase, which I'll describe next, creating two stretches of single-stranded DNA that are anti-parallel to each other. Next, DNA helicase comes in, opening the helix by breaking the hydrogen bonds between the two strands, creating a replication fork which extends bidirectionally from the origin. Following this, single-stranded binding proteins attach to the single-stranded DNA in order to prevent rewinding of the DNA. DNA gyrase comes in ahead of the break at the replication fork and helps reduce torsional strain caused by unwinding. And RNA primase comes in, creating RNA primers that are complementary to the template strand of DNA. DNA polymerase 3, which I'll draw in blue, adds nucleotides to the 3' end of the OH, or the 3' OH end of the primer strand. Because the strands are anti-parallel, one strand is synthesized continuously towards the fork, or the 5' end. This end is called the leading strand. The other strand is synthesized discontinuously away from the fork towards the 3', or towards the 3' end. The fragments that are formed as a result of this are called Okazaki fragments. This strand is called the lagging strand. DNA polymerase 1 comes along and removes the RNA primer that was added by RNA primase earlier, replacing RNA nucleotides with DNA nucleotides, moving 5' to 3' on the complementary strand, leaving what are called nicks in between the fragments, which are basically these spaces that you saw before. Say we had this and we had the spaces. DNA ligase would come in afterwards and basically fill in the space, sealing a nick by helping with the formation of phosphodiester bonds between the 3' OH of one nucleotide and the 5' phosphate end of the next fragment. This basically sums up how your replication fork would work in DNA replication.